For my students studying prehistoric life, I get to a point where I say, all right, I'd like you to think of an animal, name an animal, and we'll stop at the first 10. And so after a second, people just shout out, cat, dog, bear, person, etc. And then after, we get to our first 10, I point out that in many classes, they've only named vertebrates. And in most classes, they've only named mammals. So when you think of an animal, for many people, the first things that come to mind are not only vertebrates, but mammals. The problem with that is only 5% of animal species are vertebrates, and a much smaller percentage are mammals. Mammals certainly aren't even the most abundant vertebrates, not even the most abundant vertebrates on land. And so therefore, we have a biased view of what an animal is. And so if then we were to talk about fossil animals, here's a problem. All right, so when one say, let's talk about the history of animals or how animals could evolve. If you're thinking of these complex mammals, you're not thinking of the majority of animals. Almost all animals are invertebrates. More than 95% of animal species and more than 99% of animal individuals are invertebrates. And thus the same would apply to fossils. When one considers fossils, instead of fossils of Tyrannosaurus rex or fossils of human relatives, the overwhelming majority of fossils are of invertebrates, just like the overwhelming majority of animal individuals today are invertebrates. So if we would like to discuss fossil animals, obviously invertebrates have to be mentioned, even if they aren't the emphasis. They are the majority, so they should be mentioned. But then also, we should mention them because the complex animals, the complex mammals, say ourselves, our complexity did not appear suddenly. It was built in stages. And many of the foundations of human complexity were built in ancestral invertebrates. And so therefore, a study of invertebrates is not only valuable because invertebrates make up the majority of animals, but then also this is where the foundation of the vertebrate body plan was laid down. Right? So most of the Earth's history has 0 animals. So the Earth is more than 4.5 billion years in age, and there are absolutely no animals in the Hadean, absolutely no animals in the Archean, and for most of the Protozoic, there are absolutely no animals. So animals do not go back to the history of Earth, nor to the history of life on Earth. Life on Earth, which starts in the Archean, goes on for billions of years before the very first animal. There are some publications which state that animals have been found in the Tonian period. Others disagree on the dating. Dating soft-bodied animals early in Earth's history when stratigraphy is more difficult. It's easier to come up with alternate interpretations. The same thing would apply to the Cryogenian period, but certainly by the Ediacaran period, there are definitely animals and a diversity of animals. They are all invertebrates. So the first vertebrates will evolve in the Cambrian period, which is the first period of the Phanerozoic. So this period here is a period in Earth's history where there are animals, but there are not vertebrate animals. So invertebrates come first. There are invertebrate animals before for tens of millions and perhaps hundreds of millions of years before there are vertebrate animals. So not only are invertebrates the most common animals, they come first. Now, one of the things that makes an animal is the idea of it being multicellular. So for bacteria and for most protists, the individual is the cell. In contrast, for animals, the individual is the group of cells. We are multicellular. We are not the only ones to be multicellular. Many algae are multicellular. There are multicellular green algae, brown algae, red algae. There are multicellular fungi. All fungi except for yeast are multicellular. So here's a green alga. The individual is the group, although this, like sponges, show a lack of tissues in that. Here you have a colony, a bunch of organisms, but if you were to separate the individual cells, they would still be alive, and then they could get together and form a new colony. Some sponges can do that as well to varying degrees, where even though it's in a multicellular animal, if you were to separate the cells, they could then contribute to um, a new sponge. So multicellularity evolved multiple times. And there are these transitional phases where, unlike ourselves, where our cells could never reassemble um, and form a new organism, there are these organisms which are kind of transitional between being being multicellular and colonial. There is a group of protists. Protists, once again, are eukaryotes, which don't belong to bigger groups such as plants, animals, or fungi. This group of protists is called the choanoflagellates. And these are so similar to the main sponges the main cells of sponges. So when we talk about sponge anatomy in just a minute, most cells are called choanocytes. They have this collar, they have these flagella, and there is a group of protists known as choanoflagellates that have a very similar structure, but they are primarily unicellular organisms. Now, when I say unicellular, it is possible that they group together in small colonies from time to time, which I'll get back to. These choanoflagellates are genetically more similar to animals than any other group of protists. There are lots of groups of protists, but there are just genetic similarities between the choanoflagellates and animals. So for example, there are very important proteins in animals called the receptor tyrosine kinases, which choanoflagellates also have, unique among the groups of eukaryotes. Animals and choanoflagellates have these cell membrane proteins called receptor tyrosine kinases. So some animal features are present in this group of protists, but not enough to call them animals. And they can group together in a number of scenarios. Now, this isn't them being multicellular, but nevertheless, here we see them associating in a colony. And so it seems that, some degree of, grouping together developed in their common ancestor, and then more than evolved in the animals. So when animals appear in this fossil record, that's significant because when we look at life, all right, so yeast and even the choanoflagellates, they are not animals nor are bacteria, but all of these other organisms are, including humans, which means that they share biological traits. That's the reason that we group them together. And so, for example, they are all multicellular. They all get their nutrition from other organisms. So without help of endosymbionts, none can perform photosynthesis. None are, truly decomposers. And so here we can see a sponge under a microscope. Notice it's made of many cells. The individual is the group of cells rather than the individual cells. One of the reasons that the cells adhere is because the cells, in addition to producing protein inside the body, produce proteins outside the body or outside the cells, what are called extracellular proteins, such as collagen. Now, for a sponge, collagen is the main extracellular protein. In human bodies, collagen is the main extracellular protein, and also then the main protein of the body. So in all animals, what is the main protein making up the majority of the protein? It is collagen, whether that be a sponge, a microscopic rotifer, a great blue heron, or ourselves. And so animals have this matrix helping them to be multicellular, and the cells then group together in this matrix, unlike unicellular protists. And so animal ancestors evolved these traits, and thus they are then shared among the animals of today. Now, if we were to consider the animals of today, sponges are the most primitive modern animals, which is why it is significant that a number of papers have then published that sponges were the first animals in the fossil record, that in the Ediacaran period and even prior to the Ediacaran period, that there are fossil records of sponges. So if you were to look at a sponge under the microscope, there are no tissues here. All right, so there is no muscle, there is no neural tissue here. You can see these are large cells, but these are producing eggs. Other cells would produce sperm. But notice that they're not all in one place. All other animals have what are called gonads, where reproductive cells are made in specific parts of the body. Sponges would be the exception. Reproductive cells can be made in any place. And so sponges lacking tissues, the only animals that do, and then having these and other primitive features, they would reasonably have been the first animals, and there are fossils which have published and claimed just that. Now, sponges continue to be very important today. So, for example, they are a component of reefs. Today, they're not as important as, say, corals are, although they still are an important component of reefs. But in the fossil record, they predate corals. And so, therefore, the earliest reefs would then have had sponges as their only, and then later a major component. So you can see that reef building organisms are then important for all organisms of the seafloor, because that just provides habitat. Here, fish even today, associate around as sponges. Now, sponges are animals which feed on others, but they are filter feeding. They suck in seawater, and then they then ingest a cell at a time. The blue-green algae, the protists, etc. And so sponges, like all animals, need to ingest other living things, but they are filter feeding microscopic living things from the ocean in order to do that. Now, certainly by the Cambrian period, there are diverse sponges which are known. And so if you were to go to, say, for example, one of those Lagerstätten, one of those fossil beds which have great preservation of fossils, you can find a number of different kinds of sponges. So sponges, including ones which we can group in modern sponges, exist by the Cambrian. There are diverse kinds of sponges alive today. And I provide a number of their names. I'm sorry, the font had the outline of the letters too big, so I apologize if that's hard to read. And in addition to what are the relatives of the sponges alive today, there was an extinct group of sponges known as archaeocyathans. So this is an important point. In the fossil record, not all things appear at the same time, and then some things die at specific points. So just as the dinosaurs became extinct at the end of the Cretaceous period, and there were diverse reptiles which became extinct at the end of the Permian period, these archaeocyathans were very successful in the early Cambrian, but in the mid-Cambrian, there was an extinction epoch where the archaeocyathine group completely disappeared. Now, today, the loss of sponges would be significant, but remember that in the Cambrian, there weren't yet coral reefs. And so then the loss of a major reef-building organism would have had diverse effects on other groups as well. So in the fossil record, there have been diverse sponges, including these archaeocyathine sponges, which only lived in the early to mid-Cambrian and which became extinct at the end of the Cambrian. But then there have been fossils of sponges related to the sponges which are alive today, and sponges have survived till today and can then make various components of their skeletal elements, so including some silica. So there are what are known as glass sponges. And when we talk about different fossil rocks, like chert has silica, and so things which produce silica, which are biological, like the radiolarians, like diatoms, and like some sponges, this is important. Okay. So sponges are known in the fossil record. Oops. And I just wanted to add together just a couple of thoughts, because I had mentioned that there were some disagreements on this. Scientists don't always agree, and that is frustrating. It's certainly frustrating for me. However, that's actually the way that science works. All right, in say the Middle Ages, and prior, they agreed on everything. This is the way that the body works, or that life works, or whatever. But then the problem was they didn't constantly look for new explanations, and just ignorance persisted. I mean, they got most of it wrong, and these errors just persisted for centuries, for 1,000 years, simply because they didn't have this systematic way of going back and saying, was there really enough evidence? Can we go, check again? And so, as frustrating as it sometimes is, that scientists may disagree, or that something that was published, then someone says, wait, I'm not quite so sure about that. Nevertheless, that's how we pursue science. And so there have been fossils which have been found in the Ediacaran period, and then even going prior to that, which some have interpreted as sponges. And they've published these results, all right? And so, here I give some of the scientific, papers here. And so there have been studies saying, here's a fossil, and these are early sponge spicules. These are early sponges of body fossils. But then others have said, I'm not as sure. I would like to reinterpret that. I'm not quite sure it was, a sponge. So once again, sponges being the most primitive animals, it certainly seems as if it was most likely that they would then be the first animals, and many publications have been just for that. But just to point out, we're constantly reevaluating the fossil record, and some have proposed reinterpretations of some of those early fossils. By the Ediacaran period, a new group of animals appears, which are metazoan animals, including jellyfish relatives, the Cnidarians. And so metazoans would then not include sponges. Sponges would be animals, but not metazoan animals. The metazoans would then include jellyfish. All the metazoan animals, they truly have tissues. So in addition to just being multicellular, there are specializations where some cells will form muscle, some will form neural tissue. There will be tissues which are comparable in human embryos to the endoderm that forms, say, the gut lining, and the ectoderm, which forms the outside skin. Cnidarians don't have mesoderm, our third tissue layer, so they are called diploblastic, having two tissue layers, whereas worms and other animals are called triploblastic, having mesoderm as well. And so this means that one must regulate one's genes so that, turning on certain genes in one tissue, but not in all tissues. And when we look at these jellyfish swimming, we can see that they're doing, things that these sponges simply can't. They can move with true muscle. And I'm not being kind and generous and saying, oh, let's call it muscle. No, it's muscle tissue, composed of the same cytoskeletal elements, using the same genes to signal muscle differentiation that we use. So it's true muscle, and they control this muscle with true nervous tissue, with true nerve cells releasing neuropeptides and neurotransmitters. Now, they don't have a brain or spinal cord, and so the nervous system is organized kind of in a net, this just diffuse net-like structure. But we have that as well. Around our GI tract, for example, we have a nerve net, which arguably hails back to the days before there were brains and spinal cords. And so when we find metazoan animals in the fossil record, it's exciting, not just because jellyfish and corals, et cetera, are important animals, but then also because we are metazoans. And the body plan of the first metazoans, having genetic mechanisms to have different tissues, including muscle tissue and neural tissue, letting nervous cells conduct electricity and control the contraction of muscles. This is important for humans, but this goes back more than 600 million years to the early Cnidarians. And so finding Cnidarians in the fossil record is exciting, even if you're not interested in them, if you're instead interested in, say, the human complexity, because components of the human complexity were thus established by the point in the fossil record where we find the first Cnidarians. These Cnidarians here are called hydra. Now, they are just visible to the naked eye, but it's important to point out that when we think of animals, we often think of big things, but lots of animals are small. There are some very simple animals, which are arguably kind of in the same ballpark as sponges, which are microscopic. There are worms, which are microscopic. So animals don't have to be visible to the naked eye. And here you see a Cnidarian, a hydra, which is just big enough to see with the naked eye. Notice that it moves. It has muscle, and it controls these muscle contractions with a primitive nervous system, although it does not have a brain. It has these little arms that have stinging cells. I'm sure many of you have gotten stung by jellyfish, and I can appreciate this. These stinging cells can help trap food, which are then brought into the mouth and then into a gastrovascular cavity. Any waste is then expelled out the mouth again. So this one opening serves both as the mouth where food enters, but then also the anus through which waste is expelled. And so Cnidarians are a diverse group, including things, including hydra, sea pens, sea fans, and jellyfish. Cnidarians are first known in the Ediacaran period, where they are the most common and diverse animals. So if Cnidarians, ever had a time when they ruled the world, it was the Ediacaran period. But then certainly by the Cambrian, there are Cnidarians as well. In this next image, you can see that Cnidarians, they are a bit more complex under the microscope than sponges are. They've got different layers of cells which have differentiated. This is endoderm, which is producing enzymes, all those dark dots, for cells whose secretions are helping to digest their food. They have muscle and neural tissue. And also notice that the cells which are going to make the ova are all in one place. So they have true gonads. Reproduction isn't occurring any old place in the body, only at specific sites. Cnidarians are radially symmetric. So when you think of a pizza, You can cut a pizza in half multiple ways. You can cut it in half this way, that way. And a jellyfish would be kind of similar. You could cut it in perfect halves in multiple planes. You can't do that with, say, a fish or a gray squirrel, because the top half is not the same as the bottom half. One has the head, the brain, the eyes, etc. The other half doesn't. And so many of the higher animals are bilaterally symmetrical, that the only way to cut them in equal halves is to make left and right halves. But these simple animals are radially symmetrical. Now, radially symmetric animals, one of the issues that they then have is it can be expensive to make something like eyes. So for example, Cnidarians do have simple eyes. They're not eyes for vision per se, but more, for detecting light versus dark and for detecting shadows. Now, this is valuable because if you're trying to swim in the ocean, knowing which way is up is valuable. You can see the light. Knowing night versus dark is valuable. You may feed at a specific time. And also being able to see shadows is arguably valuable as well if other organisms are passing by. But if you're going to make eyes, they have to be primitive, because otherwise it would get expensive, because in a radially symmetrical animal, you don't have a front. Sometimes the current is, floating you this way, but sometimes it's floating you this way. Sometimes it's floating you this way. So sometimes you're going this way, that way, that way. Where would you put your eyes? Well, you kind of have to put your eyes all over the place. So therefore, they must be simple and cheap. All right, and not overly complex. One of the great advances in the bilaterally symmetric animals, is to always go in the same direction. And in that point, then you can make more complex structures. So there are fossils of jellyfish like animals which are known going back to the Ediacaran period. And then there are fossils of corals. Now, corals are easily fossilized because of their hard parts. They can make a little cup that they develop in made of corallite, which is calcium carbonate. And so as we look here, this is an ancient coral called a tabulate coral. Each of these little spaces had a space for a little coral animal. So coral animals are little cnidarians, but they can make cups for themselves. And then these cups can essentially be colonial, right? So whether it be these tabulate corals here or these modern corals here, this is where lots of little animals live. So a little animal lived right at that spot in that depression. A little animal lived there, a little animal lived there. Not all corals need to be hard. There are soft corals as well, where colonial cnidarians can live in aggregates. But the hard corals are important because they then can compose the reefs on which so many animals then depend. So coral reefs, really important. So today, for example, coral reefs do not make up the majority of the ocean floor. If you were to put all of the coral reefs together, it would maybe equal, the states of New York, Pennsylvania, and New Jersey combined. But coral reefs are critical because a quarter of all of the ocean's fish spend at least part of their life cycle in the coral reef. Coral reefs are the most diverse places for aquatic life in the oceans. So corals then are incredibly valuable for other organisms because they build these reefs, which provide the ocean's most important habitat. The reef-building organisms have changed over time. So archaeocyathid sponges, they were important reef-building organisms for a bit. Regular sponges of the phylum Porifera, they are important in reefs. Today, they still contribute to reefs, but say going back to the Cambrian, they were major constituents of reefs. There were giant clams for a bit called rudist clams, which were important in ancient reefs. But corals of the kind we have today, they have been absent for half of the history of animals. All right, and so modern corals have not always existed. If you were to go back into the Paleozoic, which is from 540 million years ago to 250 million years ago, there were no modern corals, but there were corals. They just belonged to other groups, like the tabulate corals and the rugose corals. So these corals were, main constituents of Paleozoic reefs, but the Paleozoic era ends with the worst mass extinction in the history of our planet. Continents had fused. There were the biggest volcanic eruptions in history in Siberia, known as the Siberian Traps. And during this time period, about 90% of life died out. And that included the corals of the Paleozoic. All right, it was thought that the oceans warmed. They had so much acid that it was impossible to make, the hard calcium carbonate. And so therefore, these tabulate and rugose corals, they are known only in the Paleozoic era. And it was then after this mass extinction in the Mesozoic, that is when the first of the modern corals evolved. So corals have not always existed. They've only existed in the Phanerozoic eon. And while corals first appear in the Ordovician period, they are not the corals of today. They belong to extinct groups which die in the Permian extinction. And it would then be in the Mesozoic era that we get the modern corals of today. So corals and other organisms are extremely important to all of marine life in their ability to form reefs. But the organisms which have been the components of reefs have changed over time. As I had said, corals are cnidarians. They are these tiny little animals which are carnivores. They can hunt prey with stinging tentacles. They can bring their prey into the mouth. They also then include an endosymbiotic algae. Dinoflagellate algae live inside them. That's these dark spots in their arms, so that corals also perform photosynthesis, even though they are animals. That's why they can form those reefs. They make a cup of what's called corallite, made of calcium carbonate around them. And this is what makes up the rock-hard coral reef around them. When this is preserved, it then becomes limestone. Now, there are different types of calcium carbonate. It can occur in the form of calcite or aragonite. Modern corals primarily make reefs of aragonite, whereas the fossil tabulate and rugose corals, it was primarily calcite. So once again, these reefs, and here you can see all these corals making this, rock-hard calcium carbonate. This can then become the rock which we know of as limestone. And so when we look at modern beaches, you can also often see just the hard, bits of coral, the calcium carbonate of coral. Well, once again, this can then form limestone. And a lot of limestone then actually then has the cups where the coral animals lived. And so you can actually look, here's a piece of coral, and a coral animal would have lived there and there and there. And so when you study limestone, you're learning about ancient reefs. So even if you're on land and you find limestone, that indicates there must have been a sea here at one point. There must have been a reef here where these animals living in the bottom of a shallow ocean, and they need to be shallow because they're performing photosynthesis. They made this coral, and this undoubtedly was important for the other animals of the time as well, because reefs serve, then as habitat. So much of the limestone of the world has been produced by coral reefs. And so not only is that important, limestone's important, the marble that it can be converted into is important. But also it tells us about ancient ecosystems, the extent of sea level, etc. Now, in addition to jellyfish, there's another group which are called the comb jellies. They were once classified with the jellyfish, they no longer are. And there has recently been differing opinions on how to classify these comb jellies or ctenophores. Now, one of the reasons for this is because we can compare organisms using different traits. We can use anatomical features. We can use genetic features. And sometimes the studies don't agree. Sometimes if you use these genes, it would say place the branch here that, oh, they're cousins of the jellyfish. But then other genetic comparisons using different genes might say, no, I think their branch point is a little above that point here. So I think they're actually a little closer to worms than are regular old jellyfish. One study, I said, wait, no, these might even be more primitive than sponges. Now that seems to have been contradicted, but the point is there has been disagreement about where in the family tree of early invertebrates the comb jellies belong. They are more advanced than jellyfish in a number of features. So for example, some of them have separate openings for a mouth and anal pores, but then they're more primitive than others. They're the largest animals alive today, which move only by cilia. All right, so that, there are microscopic organisms which move by cilia, but they're the only animals which move only because of cilia. There is this fossil, Eoandromeda, classified from the Ediacaran period, which seems to be a basal comb jelly. And there are a number of comb jellies which are also then known in the Cambrian. The Cambrian comb jellies are primarily stationary, which is unusual because while there are a few stationary comb jellies alive today, the majority then swim and are modal like jellyfish. And so it seems that the ancestral condition for comb jellies was to be more like a hydra, more stationary. The number of arms vary, with some fossil forms having as many as 18. And it wasn't until later that then there were the swimming comb jellies, and some of the fossils, even the cilia, have been preserved. And so I mention these at this point because comb jellies were traditionally aligned with jellyfish, although their exact point in the family tree is near the branch for Cnidarians, but there has been some disagreement among published works. And so this was a beginning of the introduction into invertebrates and their fossils, going through sponges and Cnidarians, and the next video will continue with bilaterally symmetrical invertebrates.